Hello, my name is Katherine Naasko, and I got my PhD in soil science last month from Washington State University. Now I'm a postdoc at Michigan State University Kellogg Biological Station. Today I will be sharing a snippet of my PhD research that's centered around evaluating soil health of dryland winter wheat cropping systems in the Plough soil series, using soil cores sampled to 3 feet deep. Before I dive in, I would like to thank Carol for inviting me to speak today and the larger Wazoo community for their support during my PhD, including the USDA, NRCS, and ARS for funding and access to long-term research sites. So first, let's review why soil health is important. Many soil health properties are directly impacted by soil carbon and nutrient cycling, which are driven by the soil-plant microbe continuum. Soil health assessments commonly include chemical, biological, and physical soil health properties to assess how the soil functions as a vital living ecosystem that sustains all forms of life. Furthermore, the soil ecosystem is directly connected to plant, air, and water health. However, the health of deep soils is not well studied or understood. However, in the Plough River Basin and other dryland cropping regions, plant roots extend several feet into the soil profile. As a result, it's important to study how resource gradients of plants, minerals, and inherent soil properties impact soil carbon and nitrogen pools and microbial biomass. So we took samples in October of 2018 from the Plough soil series. We sampled soil cores to approximately 3 feet deep from 3 conventional till sites and 3 no-till sites that have followed winter wheat, spring wheat, and legume crop rotations for several decades. We also sampled from a centrally located perennial grass strip that has not been fertilized since establishment over 20 years ago to compare how annual cropping systems differ from perennial systems with respect to lower chemical inputs and associated biological soil health. These 7 sites were sampled from 3 locations with 5 soil cores that were homogenized after winter wheat harvest occurred at annual sites. And these sites were also sampled across the mean annual precipitation gradient that occurs in the Plough River Basin. So soil cores were segmented into the following depths in accordance with the NRCS soil survey description of the Plough soil series. The 2 primary research questions that we asked in this sub-study were, first, how does PLFA biomass change with the soil environment, particularly with depth and across different managements, and how do these changes correspond to changes in soil health properties? So what is PLFA? So PLFA measures who's there relative to the rest of the microbial community. So today we're going to talk about a couple of main groups of PLFA, including gram-negative bacteria, gram-positive bacteria, and fungi. So gram-negative bacteria are generally faster growing bacteria that metabolize more simple carbon compounds, such as those that come from plant litter. On the other hand, gram-positive bacteria are slower growing bacteria that metabolize more complex carbon. They also have the ability to form endospores to become dormant under moisture stress. And in particular, actinobacteria are a specific type of gram-positive bacteria that can form hyphae-like branches for nutrient acquisition. Some species can also fix nitrogen, and others can decompose chitin. On the other hand, fungi are typically also slower growing and decompose more complex plant carbon, such as chitin, and can form symbiotic or pathogenic relationships with plants. So some of the other soil health analyses that we included were different carbon and nitrogen pools. So we used combustion to analyze for total carbon and nitrogen. We did some chemical extractions, including nitrate and ammonium. We also measured permanganate oxidizable carbon, and we measured ACE protein. Water extractable carbon and nitrogen pools that we analyzed included cold water and hot water extractable carbon and nitrogen. We also included a couple of other soil fertility properties, including soil pH and base cations, including calcium, potassium, magnesium, and sodium. So let's jump right in. So one of the major findings that we observed were that there was greater relative abundance of actinobacterial PLFA biomass in annual winter wheat cropping systems compared to the unfertilized perennial wheatgrass strip representing a restored native ecosystem. So this graph, you can see on the y-axis, you can see depth in inches going down into the soil profile, and then across the x-axis, you can see the general relative abundance of these actinobacterial PLFAs. And the perennial grass strip is in gray, and no-till winter wheat sites are in red, and conventional till is represented by the blue line. So, as I said before, actinobacteria are a specific kind of gram-positive bacteria. It's interesting that we did not observe these effects in the other gram-positive bacteria, so that highlights the benefits of actinobacteria on soil health in these dryland and winter wheat cropping systems. And there are two kind of notes that go along with it. The effects on these actinobacteria were more prevalent as sampling depth increased. And the other thing is that these effects were not observed in PLFA representing the other gram-positive bacteria. So using total PLFA across all the management and depths that we included, total carbon and nitrogen best predicted differences in PLFAs. So on this figure, you can see that this is an NMDS, which shows the general distribution of how PLFAs are distributed across the depths and management. So we have the depths, the different depths indicated by the different shapes, and then the different managements are indicated by the different colors that are the same as the previous slide. So pretty much the closer that 2 points are, the more similar that they are. The farther that 2 points are, the more dissimilar that they are. So when you run stats on this, the unfertilized perennial wheatgrass site was distinct from the no-till and conventional till sites. However, no-till and conventional sites were not different from one another. And then when you run the stats, all horizons or depth increments were distinct from one another. As you can kind of see outlined, has the 0 to 2 samples are these circles over here, and then the boxes with the X through them are represented over here. So as indicated by the title of this slide, variability of PLFAs was best explained by total carbon and total nitrogen, and those are kind of the furthest 2 points out of the abiotic soil properties that we included in our assessment. So on the other hand, there were some of these smaller nitrogen and carbon pools that also pretty strongly correlated with total PLFAs. So as just mentioned, the smaller fractions were less correlated. However, smaller fractions of carbon and nitrogen allow for more interpretation of biological soil health. So for example, like water extractable carbon and nitrogen indicate what's more available in the soil solution. POXY indicates the oxidizable or easily decomposed active soil carbon. And then ACE protein indicates the organic N that's considered to be available for mineralization by microbes into ammonium, which is the plant available inorganic form, which can be further nitrified by microbes into nitrate. So generally, soil health impacted biological soil health is represented by PLFA biomass more than management with respect to cropping system and tillage or climate after harvest across the Plough soil series. So this box here, you can see that depth impacted all of these kind of indicators of the different PLFAs that we observed. Cropping system impacted a couple of them as well, but tillage did not significantly impact any of these groups, and climate impacted just the other gram-positive bacteria. So this is kind of a figure just showing these relationships and how they change as you go down into the soil profile, and we can discuss this slide more later. So a recap of some of the takeaways. The winter wheat cropping systems with annual planting, fertilization, and a legume every 3 years promoted greater relative abundance of gram-positive actinobacterial biomass than the unfertilized perennial wheatgrass strip. These effects were more prevalent as sampling depth increased. Total carbon and nitrogen were stronger predictors of differences in total PLFA across management and depths than smaller carbon and nitrogen pools. However, using chemically and water extractable pools of carbon and nitrogen allows for more interpretation of biological soil health. And lastly, soil depth impacted biological soil health is represented by PLFA biomass more than management with respect to cropping system and tillage or climate after harvest across the Plough soil series. And I would be happy to take any questions.